afternoon, and I'd like to welcome you to this thematic session on precision medicine, which is generously supported by Cambridge PRISMS. I'm Alan McNair from the Chief Scientist's Office. I'm happy to be co-hosting this session with my CSO colleague Louise Campbell and Marian McNeil, who's the Chief Executive Officer of Precision Medicine Scotland Innovation Centre. Well, of course, the real stars of the show are the 5 speakers you can see behind me, who you'll hear from very, very soon. So obviously after this morning's very exciting sessions, we are running a little bit late. We'll try and push through just as quickly as possible. And I'm sure if we bite a little bit into lunch, that'll be fine for everyone as well. So really, my next slide is just really a very basic definition of precision medicine. So I think a working definition of precision medicine is that it involves using a suite of approaches to stratify an affected population, or I think based on today, the possibility of using precision medicine as a preventative approach, even more general populations, and to stratify these into clinically actionable cohorts. Why would one do this? I think it's got advantages around early diagnosis and prevention. It also allows really tailored treatments and diagnostics, and also offers the potential to really, in an enhanced manner, measure the effectiveness of interventions for various conditions. But I think it actually even goes beyond this, because the very act of stratification can lead to increased understanding of the mechanisms of the disease, which then in turn feeds back and allows new approaches to stratification. So I think you could look at this schematic more as a virtuous cycle or an engine rather than a simple linear pathway. Precision medicine also has enormous potential economic benefits. Pre-pandemic, the University of Glasgow produced a paper commissioned by the UK Department of Business, Energy and Industrial Strategy. And I think this highlighted the potentially enormous impact of precision medicine in an economic sense. Okay, a lot has happened in the world since the time before the pandemic, but I still think it's clear, given these figures, that precision medicine really is an enormous opportunity that can potentially bring great benefit to the NHS here in Scotland. It's our birthday, if you haven't noticed, that's why we're here. CSO is 50. I think a breakdown of our activities really belongs in a different talk. But I just very quickly wanted to show this slide, which just shows, certainly in a research context, how money goes out the door from CSO through direct funding of research, through infrastructure support, and through collaborations. And I think all of these have great relevance to precision medicine. The talks that you will hear today, the Precision Medicine and Scotland talks, are supported by our direct funding mechanism. As I say, our research infrastructure support goes into the NHS and means that trials and studies, including precision medicine studies, obviously can operate and recruit patients in the NHS. And finally, our collaborative way of working. Certainly, as from a few months ago, just about every single of the large NHR programs are now available to researchers here in Scotland. Of course, those programs themselves are of a scale where individual projects funded through those programs can really address some of these pressing precision medicine questions. I'm now going to pass over to my colleague Louise Campbell, who will introduce the next part of the talk, which is the Precision Medicine Alliance Scotland. Thank you.
Thank you and good afternoon. So, as Alan said, I am Louise Campbell and I'm going to talk about the Precision Medicine Alliance Scotland. So this delivers on a 2019 programme for government commitment. And the programme for government is published every year at the beginning of the parliamentary session, which is in September, and it sets out the actions that the government will take over the coming year. And this serves as a key strategic plan for the government's objectives and actions over the coming year. And it's also a good way for the government to communicate its visions and goals and also intended policy initiatives over the coming year to the public as well as to Parliament. And the Chief Scientist's Office worked closely with the Chief Scientist at the time, Professor David Crossman, to develop his vision of precision medicine and to form Precision Medicine Alliance Scotland, which then formed part of the programme for government. And this really proves the importance of precision medicine to Scotland. And the strategic aim of precision medicine is to stimulate research and delivery in the NHS of precision medicine through specific programmes of work that tackles health conditions of major importance in Scotland. And this includes diseases that disproportionately impact those at risk of socioeconomic disadvantage. So the Precision Medicine Alliance Scotland call was launched in May 2021, and in total there was 10 applications submitted from across Scotland. And these were all peer-reviewed by a bespoke independent expert panel convened by the Chief Scientist Office. And following the independent expert and lay review process, 4 projects were funded, and there was a total investment of £10 million over 4 years. We'll soon hear from the 4 funded projects that all cover a wide range of health conditions of major importance. Specifically, they focus on multiple sclerosis, diabetes, critical care and liver disease. But fundamentally, PMAS, or Precision Medicine Alliance Scotland, brings together healthcare professionals, researchers and other organisations with a joint aim to stimulate and accelerate research and innovation of precision medicine, and also improve patients' experiences and outcomes, and also strengthen Scotland's position as a global leader of precision medicine and healthcare. So we at the Chief Scientist Office are working closely with the 4 projects to ensure delivery of the project on time and target. So CSO is keen to ensure and maximise impact on both an individual project level as well as a wider programme level. We are therefore implementing a programme of post-award support where we are exploring ways to engage with the wider RD&I ecosystem to guarantee impact and maximise impact from the PMAS programme. So this includes sitting on executive committees, strategic oversight boards, engaging closely with the study teams as well as relevant policy colleagues within the Scottish Government. We're now going to hear from the 4 PMS projects, and after that I'll hand over to Marian, who will introduce the presentation from the Living Laboratory in Glasgow, and she'll also be talking about the opportunities and challenges of precision medicine. After that, we'll invite our speakers back, depending on time, and we can invite questions from the audience at the end, if that's okay. So first of all, our first speaker, Professor Ewan Pearson.
Okay, thank you very much. So I'm going to talk to you about the iDiabetes platform, which is designed to use phenotyping of patients to better improve precision diagnosis, treatment and prognosis of diabetes. So diabetes is a highly prevalent disease. In fact, the latest 2022 survey came out last week showing that we're up to 339,000 people with diabetes living in Scotland, and it accounts for a large amount of healthcare costs. It's very prevalent, particularly type 2 diabetes in the lower socioeconomic groups. It's not just a disease of sugar, it's a disease of multimorbidity. It's associated with heart failure, it's associated with liver fibrosis, and it's associated with renal replacement therapy. And I have to say, it's not a disease that we particularly manage well. We have many patients with both type 1 and type 2 diabetes who have a high HbA1c, so they're well above target in terms of their glycemic management. We don't escalate treatment until HbA1cs have got to really quite a high level. And we've shown in our data from Tayside that after metformin, which is the first-line drug used in type 2 diabetes, GPs largely prescribe at random. So there is a case for essentially a clinical decision support platform that will encourage adherence to protocols and enable a more targeted approach to care, and that's what iDiabetes sets out to do. So what I like about diabetes is the heterogeneity of the disease. Everyone who walks through the door is different, and yet our protocols tend to treat them all the same. So this is actually a slightly out of date but slightly more colourful diagram for the latest guidelines for how we manage type 2 diabetes. And you can see on the left that we now have drugs that are amazing for people with cardiovascular and renal disease. So if you have existing or high risk for cardiovascular disease and renal disease, you should receive a GLP-1 receptor agonist or an SGLT2 inhibitor. There's about 55% of people are not in that category with type 2 diabetes. And then you can see here things start to get a bit more vague, and we've got many different treatments that we might be able to offer. And it's very exciting we've got so many treatments, but as the evidence from what the GPs do shows, people don't really know what to do in these scenarios. So I'm just going to outline 3 examples. iDiabetes is actually a fairly complex platform. We've got lots of interventions we're including on this, but just 3 scenarios which show what we're trying to do within iDiabetes. And the first is around trying to predict drug response. So which is the best drug for an individual to have? So we, with an MRC project across the UK called Mastermind, have done some work using primary care data. This was largely in CPRD, where we look at differential response. And here we're focusing on differential response to SGLT2 versus DPP4 inhibitors. So you can see in the plot that there's people on the right who respond better to DPP4 inhibitors and people on the left who respond better to SGLT2 inhibitors. So clearly, if you're on the right, you want to receive a DPP4 inhibitor. If you're on the left, you want to receive an SGLT2 inhibitor. And you can develop prediction models. This is just in the form of a Shiny app that tells clinicians which is likely to be the best drug for that person. All other things being equal. This is assuming they don't have cardiovascular disease or renal disease and should be on an SGLT2 inhibitor. And what we've developed, and really led very much by John Dennis in Exeter, is what we call the Exeter 5 drug model. And they have now developed a surprisingly good model for predicting which is the best drug. So this is just showing calibration plots, showing that our prediction models perform well. And we can predict now across all of the 5 drugs after metformin, which is the best drug for an individual to receive. So what about deciding allocation of drugs with cardiovascular renal benefit to those most likely to benefit? So this is sort of shows what was on the left-hand side of that original ADA-ISD guidelines. So if you have heart failure, you should get an SGLT2 inhibitor. If you have coronary artery disease, you should get GLP-1 or an SGLT2, CKD SGLT2, NAFLD, NASH, pioglitazone, and increasingly in evidence now that semaglutide is beneficial. So how can we better identify these patients within the population? Well, one way would be to try and measure some biomarkers, and there are established biomarkers that aren't widely used for screening purposes in diabetes, although many guidelines recommend that they are done. So that's BNP and troponin for heart failure. We are incorporating a polygenic risk score for coronary artery disease, and I'll give you a slide on that in 1 second. And we're measuring FIB4 and ELF assays in the population to try and identify people with liver fibrosis. So just in terms of polygenic risk scores, increasing discussions, recent paper suggesting less useful than we had originally thought, but I really like this paper from Peter Donnelly's group in Oxford, which shows, and it's all a little bit small on my screen, but so basically this is looking at incidence of coronary artery disease on the y-axis against time. The green group are people who have a low clinical risk and a low genetic risk. The red group are the people who have a high clinical risk and a high genetic risk. The purple group are a group whose risk, they have a low clinical risk, but they have a high genetic risk. So their risk has been scaled by their genetics, and the blue group are those who have a high clinical risk and a low genetic risk, and their risk has been downscaled. So for this group of people, and this is a specific group of males aged 40 to 55, then genetics here is making a difference. And we've shown in, and this is work from Ife Mordi in Dundee, that actually in people with diabetes, the genetic risk score can help differentiate and identify people who have a low clinical risk, but actually fall into a high risk category. So the final scenario was around prediction of hypoglycemia, and what we've done here is just use data from Tayside and Fife. We have all the ambulance data, which is really unusual and unique, makes it quite challenging to replicate this. We have all the hypo call-out data, so we can take all of the data we have in the population and we can model risk of severe hypoglycemia. So we can see this now on the left for type 1 diabetes, we have a hypo rate per 1,000 person years, and then in type 2 diabetes with all the different treatments. But most importantly for what we're trying to do in iDiabetes, we can then develop that into a prediction model, which we validated. So we developed in Tayside and we validated in Fife, and we can show, predict what someone's risk of getting a hypo is per 1,000 patient years, and you can see on the people on the right and the left how different their risks are. So this man on the right, if he's insulin treated, really has quite a high risk of having a severe hypo in the next year. So what are we hoping to do with iDiabetes, and what are we in the process of initiating? So we are going to be using biomarkers such as the BMP, liver fibrosis biomarkers, prediction tools, to better enable precision diagnosis, treatment, and prognosis. We can only do this in Scotland as easily as we can, and it's not easy, we're discovering, is because of Sky Diabetes. So we have a national platform for diabetes care that captures all diabetes information for everybody in Scotland. So we're working with Nest Digital and the Sky Diabetes team to implement this. We also have a patient-facing platform, My Diabetes My Way. So any of the recommendations we're going to be making on our dashboard is going to be available to patients. So hopefully we'll get their engagement in their own health. And we're working with a company, My Way Digital Health, to implement some of their prediction models. What are we going to be doing? Well, we're going to cluster randomize a third of the Tayside population or the total Tayside population with diabetes into 3 groups. 2 iDiabetes groups. The iDiabetes Plus is the one that's going to have all of the extra blood tests, biomarkers, genetics. The middle group, iDiabetes, is just the decision support and then usual care. And we're going to randomize, and then over a course of 2 years of follow-up, we'll have all the linked data. We're working with the Health Economic Research Resource Unit in Aberdeen to evaluate the health economics. And I've only got 1 minute left, so I'm going to have to be speedy. And this is what we're doing. We're measuring C-peptide antibodies, type 1 genetic risk scores. We're diagnosing diabetes better. We're using the 5-drug model to better treat patients. And we're using all the biomarkers to better allocate people to different drugs. And I'm going to skip on. I just want to show you one slide here. No, here. So this is the dashboard. We have the dashboard developed, and we are now very much in sort of the final stages of developing that dashboard and then testing. And of course, there's a huge amount of testing we need to do before we can start using this. And we are working with Scot to obtain a medical device regulation or have our Class 1 medical device self-certification in place before we start. And we plan to initiate the pilot phase in February and go live from April 2024. My last slide. So this was the kickoff in July 2022. I couldn't find our latest slide. We had another meeting just a few weeks ago where we had 50 people in attendance. So it's a huge collaborative effort to make this all happen. Thank you.
Thank you, and if we can hold all questions until the end, thank you. Next speaker is Professor David Hunt.
Thanks very much indeed, Louise. Alan, thanks for the invitation to talk about our work today that we're doing, trying to bring precision medicine approaches to multiple sclerosis, which is a disease that I think has maybe lagged behind some of the approaches that we've seen from diabetes and in Tayside, much of which has been very informative for us as we try to bring these approaches to complex brain diseases. So the first question is really why multiple sclerosis, why Scotland? Well, MS is a major public health issue in Scotland. We have, particularly in the north of Scotland, some of the highest incidences of multiple sclerosis in the world. It is the major cause of acquired disability in young adults. But equally, over the last decade, we've actually seen really revolution in therapeutic approaches, a number of high efficacy, medium efficacy, low efficacy treatments which have been developed, all of which have differing benefit risk profiles. But our use of those is currently guided by fairly crude metrics. So just to give you an idea of this, this is some data that we have from our clinic, just seeing the number of patients that we have on higher efficacy, higher risk drugs in our clinic. And you can also see here that we've got within Scotland, the scale of the problem, here in the region that we're in at the moment, the incidence of MS is pretty similar to how it is elsewhere in the UK, which is higher than many other countries in southern latitudes. But as you go higher and higher, and this is data from Public Health Scotland from the MS incidence registry, it reaches a peak really in Orkney, where we have the highest rates in the world. But the implementation of these new drugs has really revolutionized our ability to control the accumulation of disability that occurs in multiple sclerosis. But knowing how to use these drugs and how to gauge residual activity is very much a work in progress. We use very crude metrics in clinic. So this is a very typical question that we have in clinic that we will be addressing, which is, has the disease-modifying therapy that I'm on brought my MS under control? So we have crude metrics which are based on relapse frequency, but also new lesions appearing on a scan. But that really is probably the tip of the iceberg in MS. And under that, we know that there are a lot of individuals who have got measurable activity, which can be measured either by subtle changes in brain metrics on the scan, but also increasingly from technologies which I'll show you, which have ultrasensitive detection of brain proteins within the blood, which can help us work out whether or not there is subclinical disease activity going on at the moment and help guide informed decision-making, because the prognostic potential of these is increasingly well established. So, for example, we can have a patient in the clinic who at the tip of the iceberg is stable, no clinical activity, no MRI activity, who might be choosing to continue a suboptimal therapy because actually when we look underneath and we measure and we can identify high levels of brain proteins in blood, or we can look at changes with brain atrophy on the scan, we can identify that actually they have additional ongoing disease activity which is potentially suppressible. But equally, we can prevent overtreatment. The demographic of people with MS are typically people with young families, and particularly women who are thinking about having families, who may wish to avoid being on treatment during pregnancy. And we can now add, hopefully, additional evidence to reassure people that actually if they are thinking about coming off therapy, that actually these more advanced metrics of disease activity are also clinically silent. So this is something that can be targeted to both overtreatment and undertreatment. So how we're doing that, I'm going to talk through how we have been using single molecule ELISA approaches to try and identify neurofilament in blood, and how we're going to try and bring that to the clinic, and how we've been road testing this and the predictive value within studies which have been supported by the Precision Medicine Scotland, but also show how our MRI metrics are improving and how we can bring all of this together in a way that is understandable for patients in terms of digital health and using apps and interfaces that are informed by patients and the data which we provide. So there have been a number of research studies looking at this, but the Precision MS project that's being funded here is to try and help us develop the ecosystem and help us develop the ways that we can bring these metrics into routine care in the clinic. So to give you an example, this is a technique called single molecule ELISA, which is a way of measuring ultrasensitive detection of very low abundance proteins. So there are many aspects which we can measure of ongoing brain disease, one of which is neurofilament, which is an axonal protein, which is often spilt out into the cerebrospinal fluid and has been used as a biomarker for many years, but requires lumbar puncture to detect. Now, the improvement in sensitivity of our assays, and this is a single molecule ELISA machine that we have in my lab, allows a reduction of the sensitivity, because we're detecting single molecule arrays, of 1,000-fold. So what could previously be detected through lumbar puncture and invasive procedures can now be detected in a simple blood test. So we have been looking at neurofilament in the Future MS study. This is a Precision Medicine Scotland funded study that has been looking at 5 sites representative an incipient cohort of people with MS, trying to look for predictive models which we can then feed back to patients in terms of the predictive power of some of these assays that we have been looking at. So when we look using single molecule ELISA at other brain proteins, GFAP, an astrocytic protein, or tau, there is really very little in the way of predictive ability of these proteins to predict future events. But actually, when we look at neurofilament, we see that it can help us predict relapse activity, which is one of the main questions that people have at diagnosis. And we see that patients with elevated neurofilament have a doubled risk of relapse frequency in the year following the assay. And these are patients who are not yet on treatment. Then when we look back in the cohort at patients who made a decision not to go on treatment, we can identify individuals who have worse outcomes, who maybe would have made a different decision had they had access to their neurofilament data. So we've been working with colleagues, this is my old hospital in Basel, that has been trying to take neurofilament values to be more clinically meaningful, more like the kind of centiles and Z-scores that we're used to dealing with in clinic, for example, with osteoporosis, when we look at other metrics. And we've been working with colleagues also who have been developing advanced brain metrics to try and make what are quite complex variables understandable to patients through a digital interface. So this is still a work in progress at the moment, but there is a co-design element of this where we're working with patients to help them try and understand what are quite complex brain metrics to try and identify where there are new lesions, where there are new changes on the MRI scan, how to interpret neurofilament data, but also to bring that together in a readily understandable interface that we will make available as an app for patients. So the implementation of this, where there's a lot of work that's been going on in the design phase at the moment, but will be rolled out initially within NHS Lothian. But there will be a further aspect of the study to try and deal with implementation of these more advanced metrics in populations which have higher incidences of multiple sclerosis, particularly up in the Highlands and Islands, where there is also much more geographical challenge for individuals and patients to be able to get brain scans, to have blood tests, and to be able to come and see their neurologist. So I will just on that note say that so our plan really over the next 5 years is to develop the ecosystem so that things work for people with MS. But it's important to say that multiple sclerosis is really a pathfinder disease in brain diseases. It's the first truly modifiable degenerative disease that we've developed a good, effective disease-modifying therapies for. But we are beginning to see with the advent of anti-amyloid therapies, anti-tau therapies, a pipeline of disease-modifying therapies that are coming across for many different diseases of the nervous system. So we are, within this, not only working on multiple sclerosis, but then seeing how this can be applied for diseases which are going to be very much more amenable to therapeutic intervention, particularly over the next decade, and hopefully develop Scotland as a world leader for that. So there are very many people to thank, and I know I'm short of time. And in particular to Connie as well, who's been helping manage the project as well. So thank you.
Thank you, David. Now I'd like to invite Professor Stuart Forbes, who will be talking about, or with the Centre for Precision Cell Therapy for the Liver. Thank you.
Hi. Thank you very much. So something slightly different now, liver. And also our approach, I think, as well. Okay, so We do have a problem in Scotland and in the UK generally in chronic liver disease. I think that's undeniable. That graph is out of date, and unfortunately, you can imagine it's unfortunately not come down. It's actually gone up since then. So we have an enormous problem with liver disease, mostly cirrhosis, but other sorts as well. We do have a treatment for severe end-stage liver disease. It's called transplantation, but unfortunately, the numbers we can transplant across the UK, 600 to 700, and in Scotland, less than 100 typically, unfortunately. So that really doesn't touch the iceberg of the problem. I completely acknowledge we need to diagnose early, we need to do preventative medicine. All of those things are incredibly important in society, but when it presents, it does often present quite late stage at the moment. So we have different sorts of liver disease, and the point we wanted to make is for these different sorts of liver disease, we have different biological problems, and so we have different therapeutic challenges in each one. It is small on the screen. So for liver cirrhosis, we've got a problem with scarring. For biliary disease, we have different problems. Acute liver failure again, and I'll talk you through all these. And islet transplant. Okay, so why is islet transplant on there? That's not liver disease. No, but the same cell approaches that we're using, same cell therapy, is applicable to islet transplants as well. So that's why that's included. So, in biliary disease, you often actually lose bile ducts, and you get stricturing, you get cellular senescence, and in fact, to do anything about this, we really need to replace bile ducts. For cirrhosis, we need to degrade scar and stimulate regeneration, and that's a big challenge. And for acute liver failure, typically paracetamol overdose. We have large-scale necrosis of the liver. Sometimes more than half of the liver is just necrotic material. We need to rapidly get rid of that necrotic material and turn off inflammation before we get multi-organ failure and death. So we're keen on advanced therapy approaches for all of this, because I think, as you can see, for all of these, drug approaches are really not realistic at this stage of the disease. And what are advanced therapies? Typically cell and gene therapies, and they've had absolutely dramatic efficacy, but only for rare diseases, typically single gene disorders. And what I'd like to do, and what my group would like to do, is really move those out into common diseases and make them available, and that's a big challenge. So why cirrhosis, and why are we using macrophage cell therapy? So from many years of research, we've shown that if we inject a particular type of macrophages, they hone to the liver, they make chemokines, and they attract in other cells locally to the scar region and break down scar tissue. They also stimulate very nicely to a type of progenitor cell in the liver, activate that progenitor cell and help turn those cells into hepatocytes and stimulate regeneration. And because of years of basic study, we're able to take this through into humans, into phase 1 and phase 2, which we've now completed, and now we're doing long-term follow-up of that patient group where we've treated that. And that's a really rich group to look at in terms of response and non-response. And to make the case, we can look at patient factors that determine response, but we also have an opportunity now to genetically modify the cells we give, and that's very much where I think we're going in the future. In paracetamol, acute liver failure, I've talked about the necrosis. Here we use alternatively activated macrophages, which are particularly good at phagocytosing material. They're the ones that are anti-inflammatory, and they can eat the dead material rapidly in terms of inflammation, as shown here in these preclinical models of paracetamol overdose. They can resolve necrosis within hours of infusion, and it's really remarkable to see. And the idea is to move that forward into people. And I'm pleased to say we've done that now. We've opened a phase 1 study in patients who've taken a paracetamol overdose and who have significant liver injury. It's called the MAIL trial. and we've actually recruited and treated our first patients, which is very exciting. And that relies on the knowledge of the biology of the cells that we're giving and relating them to the disease phenotype. So very precise, I hope. We're going to extend this now to preconditioning livers before we transplant them, because livers, unfortunately, the patient and the liver are in the wrong part of the country, or the surgeon is. Sometimes those livers don't get to the right people in the right time, and in that situation, the liver, unfortunately, can end up in the bucket. It's a tragedy, because that one liver non-transplanted is one person dying on the list. It's an equal relationship. But with macrophage therapy, we've got every indication in vitro that we can recondition livers, again, by being able to turn off inflammation, get rid of necrosis, and really recondition the material there. So that's something that we are developing through this funding, thank you. And the last approach I would talk about is in the biliary system, where I said we need to replace the bile ducts when they're severely damaged. Now, the other problem we have in Scotland, when we, UK-wide, globally, in fact, is for transplant now, is that the livers we get are very often fatty. And if they're fatty, unfortunately, they're non-transplantable. If we plug them into patients, those livers don't work, and it's called primary non-function. So again, they get thrown in the bin. But what we realized is that those fatty livers have very healthy, in fact, super healthy activated bile ducts. I'll explain why if you want in the questioning. But what we've been able to do is isolate those bile ducts and grow them at single cell. You can see here an organoid growing from single cells. And we've created a bank of GMP compatible ductular cells now with our partners SMBTS. And the idea is that we are going into phase 1 in patients who have got biliary damage. And the first group that we're going to target is people that have actually had a liver transplant and had a complication of liver transplant, unfortunately a common complication where they get a ductular damage and stricturing. And the idea is we can allograft them using these donor-derived cells injected into the hepatic artery to actually target the bile ducts. Again, a very precise therapy for a precise problem. And we can also genetically modify those cells to cope with some of the biological issues that they will meet when they arrive in the liver. Finally, I've just mentioned islet cell transplantation, which again is targeting the liver. It's an absolutely wonderful therapy for people with hypoglycemic unawareness, and it really is life-changing. But as a therapy, it's very inefficient. You need often two transplants, 2 pancreases, and most of the islets don't engraft the liver. Most are actually lost. And work by Shireen Forbes has shown that by using different approaches, such as applying umbilical cord MSCs to turn off this inflammatory response when the cells hit the liver, you can greatly improve engraftment and efficiency and function in preclinical models. And the idea, again, is using our platform to try and bring all of these studies through into phase 1 and phase 2, and then analyze the studies as we do it for improvement for patients. We have products in different levels of development at the moment. In fact, the macrophage is now commercialized, but we want to develop it more broadly for patients. Others are more early. And by using this funding, we will be able to really do what we need to do, engage with industry, patient support groups. The bioinformaticians look at response rates and the biology we need to change in the cells to make them more efficacious. So in my final few seconds, I can just acknowledge the broad team that are actually doing this. And this includes partners from Scottish National Blood Transfusion, as I say, an islet transplant physician, hepatologists and clinical pharmacologists. And really, you need this very diverse team to do this type of thing. Finally, I'll just say it's not cheap, and that's why it's a platform. The aim is to bring in more money and more commercial activity in this space, but the aim is to get the cost down and bring it to patients that need it, who otherwise, unfortunately, succumb from their end-stage organ disease. So thank you very much.
Thank you, Stuart. Our next presenter is Professor Nazir Lone from the Traits Project. Welcome.
Thank you very much. I'm very delighted here to present around our precision medicine project, which is, again, slightly different at the acute end, time-critical end of critical illness. I'll start with just outlining the paradigm we've had in ICU research for many years. There's been a real transformation during the COVID pandemic, and a lot of learning from us internationally from the trials we've been involved in, which we can bring to Scotland as part of this platform. I'll talk a little bit about the biology. The PI for the program is Professor Manu Shankar-Hari, and that's more his domain. And then finally, the elements of the traits platform, particularly around the data and the innovative trial design where I've been more involved. So in ICU, we have these things which sound like diseases, like sepsis, but actually they're very heterogeneous syndromes, really. And sepsis, ARDS, adult respiratory distress syndrome. We finally had a homogeneous disease in terms of COVID-19 ARDS, where we could finally do some trials in a group of patients that look very similar and actually get some results and prove treatments work. But unfortunately, that's not been the case for most ICU trials. Over the last 20 plus years, we're very good at conducting trials now in intensive care units. As you'd expect, we randomize, we follow people up, and unfortunately, over many years, we generally find no effect in these trials we've done. We often have in advance pre-specified a priori subgroups where we think there may be some heterogeneity of treatment effect, and there's been more work now done looking post hoc at biological markers identifying treatment effects that vary within different groups. But up until now, what has happened, having spent millions of pounds on trials, we then find a subgroup we think it might work in, and we then get funding for another trial within that subgroup, and then run another trial for 2 or 3 years. And that has been the story of ICU research. So we're very much looking with the traits program to transform that approach, using biology as well as setting up a trial infrastructure for Scotland. So this black box of disease, we find people who are critically ill, give them treatment, and then people pop out the other end of it, hopefully. And the approach we're really trying to learn from here is bringing together clinical features, the acuity and severity of the insult, along with aspects along that biological pathway, right from genes through to transcription and translation, to identify subgroups. And using those subgroups, randomizing on the basis of identifying these subgroups to then test treatments. So the idea of these traits, these treatable traits, so these traits ideally defined through biology, we want to have evidence in advance that these traits are already associated with poor outcomes. They're easily measurable, and this is the key bit, there are already treatments out there for particular aspects along that mechanistic pathway. So we have a reasonable supposition in advance these might actually work. So one of the traits, which is going to be one of the early traits we're launching, is the lymph-resp trait. So this is the presence of lymphopenia, reasonably prevalent in critically ill populations with respiratory failure. We find that pattern in multiple different conditions. Lymphopenia, very easy to measure. The clinical phenotype of respiratory failure, very easy to measure. It's part of our bread and butter. And there are already treatments out there, potentially along those mechanistic pathways, which are candidate treatments to bring into the trial. So moving on to the trial infrastructure itself. So we have 2 key points here of consent. The first is actually very rapidly, within a few hours, recruiting patients and consenting them to be part of the platform. So that means people then come into the platform, have a baseline blood sample taken and some very limited amounts of data. And on the basis of that initial blood test, identify whether they're eligible to go into one of the traits. If they are eligible to go in a trait, there's a further randomization step then, because that then allows them entry into one of the randomized controlled trials with an intervention. They'll be then followed up and further samples taken for the biology aspect as well. And the other key plank we'll come on to is making this as data efficient as possible through data linkage and establishing a data infrastructure and data flows from each centre. So that first aspect, finally, of moving away from these very heterogeneous conditions into subgroups of patients identified using an intelligent approach who are easily identifiable. Another key aspect we've learnt from the REMAP-CAP trial, which you might be familiar with, that was one of the large international collaborative trials in COVID, identifying treatments for COVID. So using a similar approach of a Bayesian framework and making sure it's adaptive. So that means we can bring on board new treatments, we can drop treatments if they look like they're not working early, for again, making sure this is as efficient as possible, and new traits being able to be identified as the trial is ongoing to set up a new trait and new recruitments. So there's already been a reasonable amount of work done. A lot of modelling had to be done at the outset, partly so that we could identify a predictive model based on an outcome that improves precision for these smaller trials, and also to ensure that we could establish things like what a likely treatment effect size would be, trying to optimize the timing of interim analyses. A key part of these Bayesian analyses is interim analyses, and then establishing criteria which we can drop treatments early if it looks like they are not working, or actually, again, stopping early if it looks like we're able to demonstrate efficacy earlier than as a traditional trial would have a set sample size. So I think this is, again, one of the most flexible parts of this is Bayesian frameworks bring in flexibility, but underpinned by robustness as well. And this allows these interventions, which we are trialling, to drop early on pre-specified criteria based largely on the modelling as well. For efficiency, there'll be a primary outcome reported against all of the interventions, but we also have the ability to produce treatment-specific protocols for the traits as well. The other aspect of Bayesian analyses is that you can incorporate information that is already known as you are doing your analyses. And that means that usually at the outset of a trial, normally what these priors are usually uninformative. So the assumption is there's not much known. And then as the trial goes on with further analyses, those priors can change and can become what's called informed or informative priors. And that again helps the efficiency of this particular design of trial. So the data-enabled bit, which is the bit which closest to my heart, is about leveraging the data resources we have in Scotland. So Public Health Scotland, EDRIS, used for quite a long time now, for many years, to help collect outcomes for trials in a very efficient way. We want to leverage, again, our experience of a number of the investigators in the trial have been involved in data-efficient trials, including recovery and heal COVID and REMAP-CAP. So learning from that. But a new bit here is in each of the sites to actually set up efficient data extraction and transfer to the trials unit. And the reason that's so important, first of all, this is intensive care. We have highly technical, mature environments in most units. But one of the key bits we want here is to expand out research capacity across the whole of Scotland. And we have some smaller units in Scotland where there isn't the same research infrastructure as we have in the larger units. And the real hope here is we will be able to minimize data collection burden for research staff in those units. And that enables a much more efficient process to recruit patients to trials and focusing much more on the consent aspects rather than having to collect lots of information in case report forms. We also will set up, again, learning from our previous trials on feedback to sites to maximise data quality and use as much automation as possible. So a lot of work already has been done in mapping data in each of the sites, checking its quality and seeing the case report form has been designed in such a way electronically where we'll be able to switch off modules of the case report form as we gain confidence in data. So last 15 seconds on the biology bit, much more Manu's area than mine, but the key bit here is as we collect samples, these aren't just for the end of the trial, these will be samples analysed as we go to identify new mechanisms, discover new traits then, and then most importantly, matching up with treatments. And that enables us to bring on commercial partners. We already have a number interested in the platform to be able to efficiently trial their products, obviously cost to help us, cost to recover, and roll out new treatments across the country. Thanks very much.
Thank you to all our Precision Medicine Alliance Scotland speakers. I'm now going to hand over to Marian McNeil.
Thanks, Louise. Thank you, everybody. For those that don't know me, I'm Marian McNeil. I'm the Chief Executive of Precision Medicine Scotland. Oops, hold on. Get to my slides. There we go. We're there. Yeah. So, for those that don't know, we were founded 10 years ago and very fortunately founded by investment from the Scottish Government and two industry partners. And we were set up as a consortium to be truly pan-Scotland, where we involved universities, academic institutions and the associated health boards to help inform us about how we should start. And over that 10-year period, we started with exemplar projects that then attracted further interest from industry and support from CSO, which helped founded the Precision Pank and the Future MS study that David mentioned before, and how that leads on to other studies. And that's really been our model, is we want to start things that then grow and benefit Scotland in a bigger way. We're not there to hold on to that, we then are looking for the next thing that we could support when we're doing this. What's really critical to all this is what we've built now is attracting industry investment. And I think that's really key to what's happening in Scotland. We need industry support, as Anna alluded to before, to make sure that we can drive forward and keep a pace with the global market. One of the things I did want to mention, and this is probably not very clear because I lifted it off the paper, this was published yesterday in Nature Medicine, and it's a global first. So Precision Medicine Scotland helped as a partner on this project called Tayside with Tim Kendall, Jonathan Fallowfield and many others. And the important thing here is, why is it a global first? It's the first data commons for fatty liver disease, which was called NAFLD and NASH, and has now been retitled MAFLD. I don't know if I can say MASH because it just doesn't sound right. But the important thing is here, it's trying to make no assumptions when you're looking for insights. So we're bringing disparate types of data together: genomics, electronic health record data, pathology data, imaging data, and we're allowing different researchers to bring in their data, overlay their analysis together, and bring in their own tools and look for insights so that we can see if there's new biomarkers there to be discovered, and companies are very interested in this. So if you get a chance, please do look at the paper. What are the challenges that are going forward though? I think a lot of things happen to the NHS rather than with them. I think a lot of fantastic, innovative ideas are coming out from different people, but the one plea I could make is start working with the NHS from a very early stage in your innovation pathway, and that's what we do. We want to work with them so that we're making sure we're bringing something to them that will actually make a difference. We want to improve clinical outcomes for patients, and we actually want to enable SMEs to grow and develop in Scotland. And this is a partnership thing for the whole of Scotland. One of the advantages of being this way is we've now been identified as an area of high potential opportunity. That's for precision medicine in general. So all these projects we've heard about today are being globally recognised as Scotland is a great place to do innovation in this sector. So please do communicate with anyone you feel you can. If you've got ideas that you want us to help introduce you to the right people and get things started, that's what we're here for. One of the key parts of investment that brings me very nicely on to our next speaker was that helped attract UKRI funding for a Strengthen Places bid, which was identifying an area of deprivation here in Govan that could we bring economic benefit by doing precision medicine here. And it gives me great pleasure to introduce Professor Sandosh Padmanabhan, which many of you will know. who is the academic lead for pharmacogenomics and the Digital Health Validation Lab, and has just been recently appointed the Pontecorvo Chair in Pharmacogenomics. Sandosh.
Thank you. So I'm going to give you a short introduction of the Living Lab. Whenever I meet people and tell them about the Living Lab, there are two questions. What the hell is the Living Lab and what is precision medicine? So I'll give you an outline of the thinking that went behind this Living Lab initiative, and then briefly describe a couple of projects. So a plug for the Living Lab conference next week in the same place. I'll give you the QR code at the end of the talk. So precision medicine. So this is the background, and at the bottom is a book which hopefully will come out at some point. There are a couple of problems with precision medicine, and this can be summarized with these two laws. One is the inverse care law, which means that when you look at medical innovation and medical resources, they tend to be targeted towards the more affluent, resource-rich set of the population, which means we are excluding people from the more deprived strata for these. And the converse of that is the inverse benefit law, which is we follow clinical guidelines where guidelines tell us to target the drugs based on benefit-risk ratio. So we try to maximize the benefit-risk ratio. But then over time, we tend to dilute that and get more and more people eligible for treatment. So as we scale up treatment to more and more people, you increase the risk of harm and side effects. And what that means is we have two avenues for harm. One is harm that that exposure to market forces may have on the distribution of healthcare, and the other is the harm of treating ever more healthy and healthier populations. So, and this reflects precision medicine, and this reflects some of the discussion we had in the morning about equity. But we have seen successes about precision medicine, and these have come from the cancer arena, where we have novel therapies for cancer, which come with a companion diagnostic. Now, you have two costs here, the cost of the drug and the cost of the companion diagnostic, but these have been implemented and adopted into clinical care and have shown clear patient benefit. And therein lies the strength of precision medicine, but if you want to scale up precision medicine across all common diseases, then we run into a problem, and this is a simple example. If you look at the commonly prescribed drugs, this is the percentage of patients in whom the drugs that we commonly prescribe are ineffective. And you can see almost 40 to 50% of patients do not respond to the drugs that we prescribe, and these are drugs that are in clinical guidelines and have passed clinical trials. And if you look at the drug labels, there are more than 200 or 250 drugs in FDA drug with a pharmacogenetic implication in an FDA drug label. But we haven't really utilized all this information for patient benefit. And a simple reason is that most of these drugs are off patent. So there are no profits, there are no drivers to implement these or implement more personalized treatment into clinical care. So we have a problem where when we try to translate the enormous successes we've seen in precision medicine and cancer to common diseases, there is a huge barrier. And we don't see the trickle-down benefits as we would have liked to see. So when we talk about precision medicine, we've seen in the morning about this cliff edge and the valley of death of innovation. And that same thing applies here. We have a lot of exciting science happening, a lot of discoveries happening, clinical evidence being generated, and these are generally through clinicians, domain experts, government, and patients. And ultimately, it's the triple helix. It's the clinicians, academics, and industry that partner together. But each of these three groups have their own constraints and their own, expectations for collaborating and what they wish to achieve from this collaboration. So we have a huge amount of research going on. We've got a lot of exciting science and evidence coming. But when it comes to clinical adoption, that's the cliff edge. And very few of these get adopted into clinical practice. And that's where we have positioned the Living Lab almost like a last mile solution, where we are agnostic of the discoveries. What we want to do is to find and enable or develop pathways by which we can bring a lot of these early innovations and discoveries through safely into clinical care. So that's through implementation. So we heard the innovation audit that was published in 2019, and the innovation audit clearly said that Scotland had all the necessary components for precision medicine, but the biggest gap was NHS adoption and mainstreaming. So the Living Lab was designed to address that gap, so essentially as a last mile solution for precision medicine. And this is funded through the UKRI Strength in Places Fund. So we got about 38 million from UKRI, and with partner contribution, that's come up to 90 million. And the aim is it's not top down. We are approaching this bottom up. So looking at ultimately what is it important to patients, what's important to the healthcare system, and that's about transforming the healthcare system, but mostly about economic development. And when we talk about the Living Lab, our idea, so when we applied to UKRI for this fund, our concept was the Living Lab is not a room or a lab, it's the whole Queen Elizabeth University campus. It's the whole hospital, and its collaborations with industry and academia merged in. And more importantly, we sit in one of the most deprived areas in the UK. And as I mentioned in the first slide about equity of precision medicine. So we are in the ideal place. We are a big hospital sitting in the most deprived part of Scotland. And what we want to do is to test these precision medicine implementation pathways and see how that would improve the health of all communities. And more importantly, the major deliverable of the Living Lab to UKRI and Strength in Places is not publications or papers, it's about economic growth. So we are using precision medicine and the concept that implementation of precision medicine will ultimately trickle down to the community and lead to more economic growth and prosperity. So if you look at our Living Lab objectives, so we want to improve healthcare outcomes, we want to deliver substantial savings for the NHS, support companies to commercialize and attract companies to set up in the govern area. And how are we going to establish it? We've got three pillars. So we got enabling adoption, enabling environment, and enabling innovation. So we're going to work on these three platforms, and we need academics, clinicians, and the industry working together, and we have a few exemplars. So we've got in the enabling adoption pillar, we have pharmacogenomics. In the innovation pathway, we have digital health. And finally, we have a set of projects under enabling environment, which looks at radiogenomics. We're looking at spatial transcriptomics. So it's a medley of small projects under that. The core projects are pharmacogenomic implementation and digital health validation lab. The digital health validation laboratory is because the key enabler of implementing precision medicine would be digital. So pharmacogenomics implementation, I mentioned, there are a lot of drugs. We've known pharmacogenomics since the time of Pythagoras, but we still don't know how to implement pharmacogenomics. And that's basically the problem. It's complex. And this figure shows the complexity of what we need to overcome to achieve implementation. But if we could implement, even in a small scale, pharmacogenetics that's fit for purpose, i.e. not single variant, but a panel of genetic markers that you genotype once and deliver preemptive pharmacogenetics every time a patient gets a drug prescribed, then that would be useful. And it's not just useful for pharmacogenetics in general, the whole ecosystem in which we look at the laboratory resources, we look at clinical decision support, integration into the NHS IT system, patient acceptability, physician acceptability, education, the whole pipeline of activities, then that would be relevant for any other precision medicine program that comes online. Digital health validation lab is extremely important, but it's extremely heterogeneous. We've seen the projects before, and we heard about the 4 projects just preceding this talk, and the requirements are different. We also have a lot of innovation coming through artificial intelligence, machine learning. We have digital health devices, wearables coming through. All these need to be validated, and again, for digital health and medtech innovations, there is this valley of death. People develop their devices or algorithms to a very advanced stage without speaking to clinicians or discussing clinical applications. So by the time they come to clinical studies, it's completely unusable. So we want to bring innovators and inventors and industry early on and connect them to clinicians and do the clinical studies, health economic evaluation early on. Data is important, and the biggest bottleneck, data is not a problem. There are huge amounts of data everywhere. The problem is how do we curate the data, how do we make it analyzable, how do we analyze and interpret the data, and provide, as Ewan mentioned, is the dashboard, provide data that's usable data for clinicians to make decisions. So the problem is analytical capacity, the problem is getting access to data for researchers quickly. So so through the Living Lab, just to increase capacity and also to stress test the system to find out where the potential barriers are, we are currently running this OGRE program. So we innovated a few things. So very quickly, we decided that we will try and fast track 20 projects over a 1-year period, data science project using the West of Scotland Safe Haven. The problem until now is for each project you need a separate application. And so we worked with Safe Haven, we worked with the LPAC and other committees, and we got approval with a single application to do 20 projects simultaneously. So that's helped a lot. We got the approvals done. We worked with the Robertson Center for Biocomputing to provision this data for 20 researchers. But more importantly, we use the team science approach. We need to build capacity. We need a lot of people who are able to analyze data. We need people who know how the governance part, people who can deal with data, understand data, but we also need the analysts. So we've got analysts from public health and health and well-being and other places where they have analytical expertise, but they have not worked with Safe Haven data or healthcare data. So we built a team with statistical experts, biostatisticians, and clinical trial statisticians, and created multiple groups, all working together, discussing with the clinicians. And we are now provisioned all the 20 projects. One project had a problem with data, but otherwise we are on track to produce 20 papers. The reason for 20 papers is that we want to make sure that the analysis done is actually credible and of value. So the expectation here is, as the digital validation lab scales, we'll be having a pool of analysts who can manage and deliver on the heterogeneous set of projects that we are expecting to come to us. And there is an e-poster on OGRE, and there is an OGRE website which gives you details of the projects which range from cancer through to net zero to homelessness. So it's a wide variety of projects. We are using our Safe Haven data to try and interrogate. And finally, the other example I mentioned is about digital devices. There are a lot of engineers. There are engineers, very clever. They build devices, they invent devices. But the problem is most of them are solutions in search of a problem. And one of them is this group. They came to us. They've been working for many years using a laser or using light to try and detect falls in the elderly. And this was a clever device where they have a laser which is just like a laser pointer, so it's human safe. Shine the laser on the neck, that small reflected signal, they can deconvolute into sound. And essentially, they could pick up the first and second heart sounds, and they could even pick up the third and fourth heart sounds from just reflection from the neck. And they were using it to detect faults. So they came to us just to have a chat, and we suggested, oh, why don't you, instead of just detecting faults, why don't you just move it to the chest and detect heart murmurs? So they did some exploratory work, and they found they could detect heart murmurs. So we're now about to start a clinical study, 20 patients from the heart valve clinic, select them, and just validate, or the first proof of concept that this can discriminate between different heart murmurs. It's a contactless stethoscope. The first time, all the stethoscope now is with contact or is acoustic. This one is light converted to sound. And whilst we were discussing this with the engineers, suddenly the vets came to us and said, this would be very interesting for us because the horses don't like being touched or auscultated. So when you finish this, come to us and we would like to use them. So the other project is radiogenomics. This is combining imaging, transcriptomics together to try and predict risk and stratify patients, and that's led by Kevin Blythe. And thank you. This is the last slide.
A huge thank you to all our speakers. I think that was a fascinating whistle-top tour around the world of precision medicine. We actually brought that in on time to the minute, but I think the less disciplined sessions before ran over a bit. So I would suggest I think everyone probably does need a sandwich, so we probably won't stop for questions. But any questions you may have for any of the speakers, please email them via the conference website or via the CSO website, and we can get them to you. So I'd just like to finish by giving a huge round of thanks to our 5 wonderful speakers. Thank you very much.